Hello guys, and welcome back to my YouTube channel. So today, instead of going into subtopics and all that, I will be presenting a journal that I most recently read and presented to my science fair mentor. So the name of this journal was Tamoxifen Resistant Breast Cancer Cells Are Resistant to DNA Damaging Chemotherapy Because of Upregulated BARD1 or BARD1 and BRCA1 or BRCA1. This journal was published in a journal magazine named Nature Communications, and it is under the family of Nature, and this whole family is a very reputable journal source, so I had a great time reading it and learning a lot from it. And so before I go in to explain the actual paper itself, I wanted to add some backstory to help make understanding this journal a lot simpler for you guys. So my focus audience for today, I guess, is more towards hormonal breast cancer patients, especially for those that have had a recurrence and the cancer was deemed tamoxifen resistant. So the whole reason why I chose this journal and read it was because my mom actually kind of was very similar in the ways that they described this whole process. Because when my mom first got cancer, she also did tamoxifen, but then it later came back, and then she was to do she took and is taking currently a drug called Alpelisib, which is one of the drugs that is used in this journal. And I found it very interesting that a journal published 2 years ago could have so much similarity to what I am currently facing with my mother in this year. So when a breast cancer patient gets a diagnosis that they have hormonal breast cancer, after the surgery, people, or before surgery for some people, they will get chemotherapy. But alongside with chemotherapy, they will usually get a hormonal therapy, which is also a target therapy specific for the hormonal positive breast cancer groups. Of the hormonal therapies there are in this world today, tamoxifen is a very widely used specific drug for hormonal positive breast cancer patients. And what tamoxifen does is that, as you can see in this photo, it will bind to the estrogen receptors, mimicking that of a estrogen, and then later kill that cancer cell. So if it works, it obviously is very great. However, in the few unlucky cases, a cancer cell can develop resistance to the drug. And in that case, the drug, it does not work and does not do its function in killing the cancer cells, making the cancer cells still live within the body. So this paper includes a lot of specific terms that I thought would be helpful if I included it for you guys, so that you guys have a easier time understanding what I'm trying to say as I explain the figures later on in this video. So first, there are two genes that are even mentioned from the title, BRCA1 and BARD1. So what these genes are that these genes by itself are known as tumor suppressors. And BRCA1, also known as breast cancer gene 1, is also known for its role in DNA repair. So if these BRCA1 BARD1 has a mutation or a change within that is not supposed to happen, then it can turn into an oncogene. So what exactly is a tumor suppressor and an oncogene? So to explain it quickly and simply, if there is a car can be kind of deemed as the cell. And then to move the car forward, you have the accelerator, and then if it's going too fast, you have the brake to stop it. However, the accelerator can be seen as the oncogene, or the cancer-causing genes. A tumor suppressor, or the brake, can be seen as a stopping point and can just be used for blocking any bad thing from happening. And within our bodies, we have oncogenes and tumor suppressors already. And with this information, you might wonder, why in the world do we have oncogenes in our body when it causes cancer? And can't we just get rid of it and stay cancer-free forever? And all these questions. But ironically, an oncogene does not only have bad functions, but it also has a good function. For instance, when a baby is growing within the mother's womb, during the stage when it's like an embryo and it's just developing and differentiation is happening, oncogene actually helps with this whole process. So it's not a completely bad thing. However, if it gets like overexpressed or just too worked up, then it will become a bad thing. So in the title, I mentioned about the tamoxifen-resistant breast cancer cells. And as I mentioned before again, tamoxifen is a hormonal positive breast cancer patient-specific drug that is supposed to kill the cancer cells. So then why does this resistance happen? And to this question, I actually cannot give a definitive answer because each researcher and each paper has a different opinion on how they think tamoxifen resistance happens. However, in this journal that I will be presenting to you today, they think that tamoxifen resistance is caused by a PI3K AKT pathway and cyclin and CDK pathway and all these other cell growth related pathways that are trying to overcome the suppression that happened due to the tamoxifen killing off a lot of the cancer cells. So now let's get into the figures, and I will not explain all the figures like with great detail because then this video will be like 30, 40, 50 minutes. However, I will try to grasp like the main points of the figures, and I will try to very briefly summarize each figures. In figure 1, the first breast cancer cell line that can be seen is MCF7. And the reason why MCF7 is used is because it is a very well-known hormonal breast cancer cell line. In figure 1b, BARD1 is among those genes that are highly expressed in tamoxifen-resistant breast cancer cell line. And the other parts of this figure is trying to show that BRCA1 and BARD1 are highly expressed in only the tamoxifen-resistant breast cancer cell line. In figure 2, 3 chemotherapy agents are used: cisplatin, adriamycin, and paclitaxel. Cisplatin and adriamycin has a gap between their cell viability for the MCF7 hormonal breast cancer cell line and the tamoxifen-resistant MCF7 breast cancer cell line. Paclitaxel, on the other hand, has a pretty similar result for the MCF7 hormonal breast cancer cell line or the tamoxifen-resistant MCF7 hormonal breast cancer cell line. And the reason for this is because cisplatin and adriamycin are DNA-damaging chemotherapy agents, while paclitaxel is not a DNA-damaging chemotherapy agent, but rather a mitotic chemotherapy agent. And then for figure 2, the remainder we're trying to see the different DNA damages that happened. So for instance, there was like the comet assay and the foci, and then there was also like the apoptosis assay that was trying to see for like the cell death. And all of these show that the cisplatin had a high DNA damage and cell death for the regular hormonal breast cancer cell line for MCF7, but it didn't have that much DNA damage or death for the resistant breast cancer cell line. And what exactly this DNA damage and DNA repair thing is in simple terms, it can be thought of as a house on fire, as a troubled cell. And then when a house is on fire, people usually call 911. So then like the ambulance, police, fire trucks come. And so these police, fire trucks, and ambulance can be thought of as the DNA repair genes. So the DNA repair genes include ATM, HER, CHEK1, CHEK2, 53BP1, RAD50, gamma H2AX, and very interestingly, BRCA1. So this figure just in general shows that the chemotherapy for DNA damaging agents had no effect and had a slight, like really less effect than on the resistant breast cancer cell line compared to the MCF7 hormonal breast cancer cell line. In Figure 3, by using experimental methods, the researchers got rid of BARD1 and BRCA1 and saw that the reduced amount and expression of BARD1 and BRCA1 made the tamoxifen-resistant breast cancer cell line to be more responsive to the DNA-damaging chemotherapy agent cisplatin. In Figure 4, they basically did the same thing as Figure 3, except with PI3K inhibitors. So before I get too far along with that, I'll explain some of these figures. So in Figure 4a, they were trying to find out which pathway this BRCA1 and BARD1 could have come from. And of them, they found the phospho-AKT. And AKT is a well-known cell proliferation helper, and it was highly expressed in the tamoxifen-resistant MCF7 hormonal breast cancer cell line. And to check, they used the PI3K inhibitors because the PI3K and AKT are all in the same pathway, and found that BARD1, BRCA1, and phospho-AKT levels all showed a significant decrease when the inhibitors were put in to the tamoxifen-resistant hormonal breast cancer cell line. And to be more specific, the BYL719 is the Alpelisib, and the BKM120 is the Buparlisib. The Alpelisib is a PI3K alpha inhibitor, while the Buparlisib is a pan or all PI3K inhibitor. And through this, they showed that, again, when a PI3K inhibitor was added to the tamoxifen-resistant MCF7 hormonal breast cancer cell line, the cisplatin was able to do its function correctly and kill the cancer cells effectively. So Figure 5 is a preclinical study, which means that it was done on an animal, and in this case it was done on a mouse. So the tumors in Figure 5a were grown within the mouse, and how it happened was that the researchers would technically grow the cancer outside and then put it into the mouse's body by sticking it into the mouse's side. So in the first figure 5a, MCF7 hormonal breast cancer cell line can be seen as a big amount, and then the MCF7 hormonal breast cancer cell line with just cisplatin showed a significant decrease compared to the MCF7 no treatment. And then when the BKM or the PI3K inhibitor was added alongside the cisplatin, not that great of a change could be observed between the cisplatin with the only cisplatin and the BKM plus cisplatin for the MCF7 hormonal breast cancer cell line. Now, comparing the MCF7 resistant, so like tamoxifen resistant breast cancer cell line to the regular breast cancer cell line, the resistant breast cancer cell lines can be seen to be like a lot bigger than the normal ones. So in the MCF7 resistant cell line, when there was no treatment, obviously it was a really big tumor. And then when the resistant cell line was alongside with cisplatin, there was no change observed between the resistant no treatment and resistant plus cisplatin. However, when the PI3K inhibitor was added, there was a significant decrease for the tamoxifen resistant MCF7 breast cancer cell line compared to the MCF7 resistant breast cancer no treatment and the MCF7 resistant plus cisplatin. And then an IHC, also known as immunohistochemistry, was done. And once again, the BRCA1 and BARD1 levels were shown through, protein levels were shown through this specific study. And in the MCF7 hormonal breast cancer cell line, not that great of an expression can be seen in the MCF7 hormonal breast cancer cell line, no treatment, plus cisplatin and PI3K inhibitor. However, in the MCF7 tamoxifen resistant breast cancer cell line, a high amount of BRCA1 and BARD1 can be seen. And then even with the cisplatin, a greater and a lot big amount can be seen for the BRCA1 and BARD1. However, when the PI3K inhibitor was added, the amount of BRCA1 and BARD1 decreased, and through the tunnel staining, this shows like the cell death, and the darker it is means the more cell death. And as you can see with the MCF7 tamoxifen resistant breast cancer cell line plus PI3K inhibitor and cisplatin, a darker shade can be seen compared to the other breast cancer lines that is shown in figure 5C. Through this, the researchers are trying to show that the PI3K AKT pathway controls the BARD1 BRCA1 gene levels, which also controls whether or not these tamoxifen-resistant breast cancer cell lines are resistant or not to the DNA-damaging chemotherapy. This is the final figure of this journal. So in figure 6, the researchers sought for the IHC of the primary and the metastasis breast cancer cell lines. And by metastasis, they are talking about the recurred tamoxifen-resistant breast cancer cell line. And as you can see, the phospho-AKT, BARD1, and BRCA1 levels were all higher in this recurrent tamoxifen-resistant breast cancer cell line compared to the primary breast cancer cell line, which means that it is like the first breast cancer that is found like within the breasts. And then it shows that BRCA1, BARD1, and the phospho-AKT had a positive correlation. And finally, that not only can this be used in regards to chemotherapy,
but that the high BARD1 and BRCA1 expression results in resistance to the DNA-damaging therapy such as radiation as well as chemotherapy.
So thank you for watching, you guys. I hope this journal gave you and your family hope as much as it gave me and my mom hope. Because my mom, she takes the Alpelisib, which is the other PI3K inhibitor drug that this paper mentions. And although this journal did the other drug, my mom, that is currently taking Alpelisib, has shown great results. So, like for instance, my mom had a recurrence in 5 different areas, and when she did her PET scan not that long ago, 4 of the areas were completely gone, and the one area that was left showed a great decrease in size, which was very, very happy for the all of us. So I just really hope that this can become a sign of hope for you and your family if you have the tamoxifen resistant breast cancer, and that through other papers and through other research you'll be able to gain more hope for life. So in regards to these PI3K inhibitor drugs, I was thinking that these drugs, if they are easily affordable, that'd be pretty great. However, these drugs are not only expensive for some people, but because they are drugs, they have their side effects, and these side effects are sometimes really, really great. And for instance, my mom has a little bit of nausea, and yeah, so side effects are a huge deal when it comes to drugs. So I was wondering if there were any foods that we could eat beforehand or during to work as a PI3K inhibitor for the tamoxifen-resistant breast cancer patients. And here is a list of foods that I found, and I hope that you and your family will be able to eat this, and it will just be a healthy family time for all of you guys, and a hopeful family time for your family as well. So finally, each journal, and this journal had questions that I wanted to solve more and parts that I wanted to experiment more on. And I hope one day that we'll be able to work together in this in the future as we one day hopefully meet, because my mentor and I were just talking about and discussing about this journal not that long ago, and he and I were talking, and we were just like, oh, there are some parts that we could do more research on, and maybe it will become a more sign of hope for these breast cancer patients. And I just thought it'd be really cool if maybe in the future you guys and I will be able to meet and we'll be able to research together for all of this. So thank you for watching again, and I really hope that you'll come to see me again.